Looking here, we have topic 4, inheritance and variation of traits. For each section, I've listed the standard. When you're looking at the standard, you want to understand how that's going to apply to the questions that are asked. Most of the questions are going to have either data, a passage, or a model that you need to evaluate. I have tried to take the models off of some of the recent exams, so therefore you are more familiar with them in order to see what the expectations are. HSLS 1-1 says, construct an explanation based on evidence for how the structure of DNA determines the structure of proteins which carry out the essential functions of life through systems of specialized cells. Whenever it says evidence, that means that somewhere in the passage or somewhere in the model, you're going to have to grab information and use that in your question. Remember, a lot of these, though, also require you to have background knowledge, and that's where this review portion comes in. You need the background knowledge that you're going to apply to the new evidence that they are providing to you. The first part says all cells contain genetic information in the form of DNA molecules. If we think back to DNA, remember we can either look at it on the chromosome, and right here, this is going to be a chromosome. We get one chromosome from our mom, and then we get one chromosome from our dad. And if we look at the structure of DNA, remember that's going to be that double helix, meaning that it's two-sided, and helix refers to the fact that it's a twisted ladder. Genes are regions of the DNA that contains the instructions or the DNA base sequence that code for the formation of proteins. Remember, my DNA base sequence is referring to my letters A, T, C, and G. We have our base pairing rules, apples in the trees, cars in the garage. So I need to remember that A and T pair up and then C and G pair up. When we're looking at a gene, we're talking about just a tiny segment or section of the DNA code. So we're not looking at the whole thing, we're just looking at one little section, and that gene, which is a series of letters or a sequence of letters, is going to code for a protein. So the section I have highlighted here is a gene. That gene makes a protein, and that protein is going to determine what your trait is. Sometimes you'll see information about an allele. An allele is a version of a gene. If you think back to the state lab we did about lactose tolerance, we had our capital L and our lowercase l. Both of those represented different alleles for one gene. In the example I have here, I have the dominant allele, which is capital T. Dominant is the bossy one. The bossy version is going to be the version that shines through. So if capital T represents tall and little t represents short, people could have the genotype of big T, big T, big T, little t, little t, little t. Remember, my capital T is going to be my boss. So big T, little t, both of those people are tall. If you have two little t's and you don't have the bossy big T, you're going to be short. Allele is a good vocab word to know. So a gene is a segment of DNA, and then an allele are the different versions of that same gene. If I was going to memorize one thing for all of my genetics questions, I would make sure that I knew this flow chart. This is going to help you answer any question that's based on making proteins. If I see a question that's asking about DNA base sequence, amino acid sequence, protein structure or function, I know that if I write down this flow chart, I have a much better chance of getting the question right. So again, here I have my DNA base sequence that refers to my letters. I have my amino acid sequence. Remember, amino acids are going to be my building blocks of proteins. Very similar to what we did with our beads and our pipe cleaners. We did the DNA, we transcribed and translated that into our amino acid sequence, which were our beads that we put onto the pipe cleaner. After we had put all of the beads in the pipe cleaner, we then arranged it into a shape, which is our protein structure. Structure and shape for us could be synonyms for one another. The structure or the shape is going to determine the function, and then finally function determines whatever trait you have. Next up, we have transcription and translation. Understand how the DNA code is transcribed and translated in the synthesis of proteins. Synthesis is one of those words that we need to know. Synthesis means to build, to make, or to produce. And something I like to do is when I see a word that's a big word that I don't use in my everyday life, I like to switch it up for the more common words that I am familiar with. Protein synthesis has 2 steps. The first step is called transcription. Transcription has a C in it, and I remember this is when I'm going to make my copy of DNA. My copy of DNA is called messenger RNA. That all occurs inside of the nucleus. So if I have a cell, my nucleus is the brain of the cell, and that's going to be where my DNA is. Remember, DNA is a double helix, so it has 2 sides. It is very, very long. It is trapped inside of the nucleus. I don't want my whole DNA strand. I just want to make a copy of one gene. It's like making a copy of just one recipe. So the first thing I drew, my double helix, was DNA. And then I transcribe it. Transcribe means make a copy. And what I transcribe it into is known as messenger RNA. Now, messenger RNA is a copy of just one gene. After this is copied into messenger RNA, it goes to my ribosome. So now the messenger RNA is in the ribosome. The ribosome is actually going to be where that protein is made. And I think ribs are made of protein. That messenger RNA gets read in 3 bases, much like how I have here, AGC, UUU, ACG. Each one of those 3 bases is referred to as a codon. As it gets read, that is going to code for a specific amino acid. And those amino acids are going to be bonded together. When those amino acids get bonded together, they form a protein. The protein then folds into its specific shape or structure, which is going to determine its function. When the amino acids get bonded together, that's known as translation. So here I have my DNA goes to RNA. That's called transcription because I am making a copy. And I follow those base pairing rules, which is A to U, T to A, and then C and G pair up with each other. My next step is when mRNA goes to the amino acids. This step is known as translation. And you'll notice here I'm using a different language, not my bases. Since I'm now using the amino acid language, that's called translation, going to that new language. Now to review how to use this chart, we're going to start with A, G, C. These in the center are representing my RNA. I start off with my A. Notice here it says read from the inside out. So the first one I'm looking at is A, as depicted here. That means I know that that one is going to be somewhere in this quadrant. My second letter in this case is going to be G. Again, I have A, and then my second letter is G. Now I know I'm going to narrow it down to this smaller section of just this quadrant. The last one I have is C. Here I now have A, G, C. That gives me SIR. SIR is an example of an amino acid. When I bond all of my amino acids together, that is going to be when I get a protein. Each amino acid has a different charge, positive, negative. It could be hydrophobic or hydrophilic, and that's going to determine the shape that the protein makes. These are specific types of proteins that we've talked about throughout the year. We have an enzyme. The one we referenced was lactase. Lactase is the enzyme that is necessary to break down milk sugar into its building blocks. Enzymes speed up chemical reactions. They can either digest things, like in the case of lactose, the milk sugar, or they can synthesize things, like in the process of photosynthesis, where you're building or making food. After that, we have cell receptors. Cell receptors are located on the cell membrane of cells, and they act like little antennae that match up with chemical messengers known as hormones. Hormones we'll talk more about when we review our reproduction section and also human body systems. We've got testosterone, estrogen, and insulin. They get released into your bloodstream, and then they target specific cells based on which cells have which receptors. And the last one that we have listed are antibodies. Antibodies are proteins that your body makes when they are exposed to a germ. Those germs could be things like viruses or bacteria. Your body also may make antibodies when you're exposed to a vaccine or if you have allergies. When someone has allergies, their body is overreacting to a non-harmful or a harmless cell. And the thing is that those cells that are harmless still have ID tags on them, and your body thinks they don't belong and therefore fights them off. Over here, we have the process of both mitosis and then differentiation. Remember, you could see the word mitosis or you could see the word mitotic. They mean the same thing. So here I have mitosis and I have mitotic. When I see that word mitotic or mitosis, what I'm thinking is my two identical cells. So looking here, I see 2, and I remember, I make 2 cells from this. The other thing that I like to do is circle the I. When I circle that I, that helps you remember that they have identical DNA. Identical means they have the same exact number and types of chromosomes. Now going back up here to this part, this is looking at reproduction, specifically sexual reproduction. You'll see we have sperm and egg coming together. That process is known as fertilization. Fertilization restores the chromosome number. That forms a zygote. A zygote has a full and complete set of chromosomes. Now, we don't just stay as a single cell. Therefore, we are going to undergo cell division. The type of cell division that we're going to undergo is known as mitosis. That means that regardless of what cell we're looking at in humans, every single cell in your body has the same exact DNA. So once you form this one cell, it divides, and it divides forever. Right? Mitosis. All of these cells are identical to this cell. All of these cells are identical to this cell. Now you might be thinking, well, I see I have these 3 cells. Those all look different to me, and you just said they're all identical. Well, that is a process known as differentiation. Differentiation is talking about the fact that different genes are turned on. Now, that does not mean that they have different genes. It means that they all have the same genes, the same exact genetic code, just certain sections of the DNA code are turned on in skin cells, and different sections of the DNA code might be turned on in muscle cells. So differentiation is when cells get their specialized or different jobs due to genes being activated. And when you activate something, that means you turn it on. The most important thing to note is that all these cells have the same DNA. Because every single cell in your body was made through the process of mitosis. This is showing us a good example of what mitosis is. We have a parent cell that has 8 chromosomes. When it undergoes the process of mitosis, it produces 2 identical daughter cells. They're identical because they have the same number and type of chromosomes. That parent cell has 8 chromosomes, and then each daughter cell also has 8 daughter cells. Mitosis is used for 2 things: growth. Every single person starts off as 1 cell, and then the way you get bigger is through the process of mitosis. And then the other way that mitosis is used is for repair. Let's say that you go and you cut yourself and you need to replace those cells. That process that replaces the cells is known as mitosis. And let's add over here that it's going to be the same number and types of chromosomes. Now to review differentiation, we're going to look at how different sections of your DNA are turned on and how that determines what the job of a specific cell is. You have many different types of cells. In this example, you'll see that there is a neuron. A neuron also is called a nerve cell. And then we have an epithelial cell, which are going to be cells that, let's say, line your digestive tract. Every cell starts off in your body from one cell. This one cell is referred to as a zygote. Those cells divide, 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 divide. That whole process of dividing is mitosis. When I see mitosis, again, I'm thinking, oh yeah, these cells are identical. They have the same exact genetic information in them. Why, however, does one become a neuron and one become an epithelial cell? If I'm highlighting a similar area of DNA, what I'm going to notice, let's say if you are a nerve cell or a neuron, this section of DNA is activated. Remember, activated means turned on. When I flick the light switch on for this gene, that makes this cell differentiate or get the specialized job of being a neuron. If I look at an epithelial cell, what I will notice is a different section or segment of DNA is turned on or activated. Since a different section is turned on, I now have a cell that has a different function. So the big idea here is that differentiation is when different genes are activated. When those different genes are activated, that's going to determine the job of your cell. And the last thing I have listed, I think they might use as a phenomenon, potentially in the future. And it's talking about if your cell division doesn't happen as it's supposed to. Mitosis is supposed to make two identical cells and then pretty much stop the cell cycle. And then when you need other cells, it undergoes mitosis again. Sometimes what can happen is you have uncontrolled or abnormal cell division. This leads to cancer. So it's mitosis, but instead of stopping at the checkpoints, it just keeps going and going and going. And that leads to the development of tumors. When we're talking about differentiation, we also want to keep in mind stem cells. Stem cells are a biotechnology. These are cells that have no job yet. There are different types of stem cells. This image or model is focusing on embryonic stem cells. Embryonic stem cells come from an embryo. That means that these cells can become any type of cell. They can become any type of cell. Because as of right now, none of their genes are turned on or activated. So this embryonic stem cell cluster could become muscle cells. They could become intestinal cells. They could become liver cells. Our next standard is HSLS 3-1. Ask questions to clarify relationships about the role of DNA and chromosomes in the coding instructions for characteristic traits passed on from parents to offspring. The question asking can be difficult or challenging at times. You want to ensure that the question that they provide you in the multiple choice, that it is actually answering the multiple choice question that they have provided to you. You're gonna need background information in order to do that. First up, we have each chromosome consists of a single very long DNA molecule. I already drew this, but remember, we're looking at that double helix. And when I look in this section here, that latter section, that represents my letters. Each chromosome is a particular segment of DNA. The instructions for forming species are carried on that. Over here I have a picture. Notice I have 21 chromosomes. Over here we have our sex chromosomes. I don't think you'll see this, but XX is female, XY is male. Here they're only showing us one chromosome in the pair. Keep in mind that we have 2 chromosomes per pair. The other thing to note is that each one of these bands represents a different gene. Also keep in mind that when we have chromosomes, this is not a totally accurate representation. Humans have 46 chromosomes. Now on to the actual part that is really the standard. Distinction between coding and non-coding regions. We have coding. Coding codes for a protein. On our last page where we did transcription and translation, and I said transcription makes a copy of the DNA into RNA. RNA brings the message to the ribosome, and then there in the ribosome, the amino acids get linked together and make a protein. Those are for DNA sections that are referred to as coding DNA. So coding DNA codes for a protein. Our non-coding section, they don't code for a protein. They are in charge of gene expression. They're like the light switch. Looking here, I took this from the lactase lab. Here it says gene transcription occurs. When I see transcription and when you see transcription on the test, we're going to remind ourselves that this means copy of DNA. Right, the DNA goes to RNA. If you make RNA, that means that you can make a protein. Right here, this lactase, that's my protein. This DNA base sequence is my coding region. Now on the left-hand side, you'll see it's called control region. I don't know why they call it control region in this image. And then in the standard, they call it a non-coding region. You are going to have to be familiar with both of those terms. So here's my coding region, the actual gene base sequence. And then over here, this is my non-coding region or control region. When these two interact, this is going to be like my light switch for this example. So when these two interact, it then turns on this gene. If one of these regions is messed up, has a mutation, they don't interact properly, then this does not get turned on. The next page, I also took these images from a more recent Regents exam. Methylated and unmethylated have been on a few of the exams so far. For methylated DNA, which again, they give you this image, they tell you that these are the methyl groups. Might your picture look different come Regents Day? Yes. Might they not ask this? Yes. But they have asked it on a few of them so far. Here are little tags that are on my DNA. When I have methylated DNA, I have no transcription. Now keep in mind, when I see that no transcription, the first thing that I do is I'm like, oh man, what does transcription mean? I remember transcription means to make a copy. So this means no DNA copy. If I make no DNA copy, that means I make no protein. So no DNA copy means no protein. Again, circling that C seems like a silly thing to do, but when you get into all the really confusing language inside of those questions, that's going to be real helpful. Then we have unmethylated. Notice none of those things are there. Nothing's there. And here it tells me transcription occurs. If transcription occurs, I make my DNA copy, which is messenger RNA. And if I can make my messenger RNA, then I can make my protein. Next standard is HSLS 3-2. Make and defend a claim based on evidence. Evidence means that it's going to be there. It is going to be somewhere in the passage or in the picture or in the data. And you're going to have to use that evidence in order to say a claim is valid or invalid and explain why. It says inheritable genetic variations. When I see that word variation, because sometimes my students struggle with that. When I see the word variation, I just think differences. And again, if the regular vocab is what is really the stumbling block for you, I would make sure I go home and I make a list of the vocab words that I do not know. And they don't have to be science vocab words. They could be the vocab words that are on the front of this packet. Because I went through all of the tests and I tried to make a list of the words that I think maybe students will not know. So I've got one, because this actually has several sections for this standard. It says new genetic combinations through meiosis. You have to think back, what is meiosis? Well, she was just talking about mitosis. No. Those are two separate things. When I'm thinking about meiosis, I have my own little trick for that too. Mitosis, I was thinking two, identical. Now I have meiosis. Meiosis is referring to sex cells. So every time I'm looking at meiosis, I'm thinking sex cells, sperm and egg, sex cells, sperm and egg. And again, try to keep vocab clustered together. So if you are making flashcards and you have meiosis and sex cells, I'd also add the other words that we need to know, like sex cells. Those could also be called gametes. What are gametes? Gametes are sperm and egg. So I've got my sperm. I have my egg. Up here I have a more formal definition. I like trying to keep it simple though, clustering those words or vocab together to help me remember. Meiosis is a process of cell division that produces gametes, which contain half the normal number of chromosomes. This process creates 4 unique gametes, increasing genetic variation to the next generation. You need to make sure that sperm and egg only have half the normal number. Remember, half the normal number for humans is 23 chromosomes. So here, when I say sperm, Sperm has 23. Egg has 23. On the last page, I said a full and complete set for humans is 46. So when you put those two together, 23 plus 23, you get 46. This half the normal number is important to ensure that chromosome number stays the same for every generation. Write that down. The other thing to note, right up here, it talks about meiosis increases genetic variation. Variation is differences. This is why you and your siblings are not exactly the same. Each sperm and egg are unique, right? Even if your parents make millions of sperm, millions of eggs, every single one of them is going to be unique. So unless you are an identical twins are formed from the same sperm and the same egg, you will not have the same DNA. You will have similar DNA, but you will not have exactly the same DNA. So this is why siblings are different. Each sperm and egg is unique. Another reason why each sperm and egg are unique, right over here, we've got something called crossing over. Crossing over, when I'm looking at it, my chromosomes during meiosis, they get real close to each other. When they get real close to each other, these sections intertwine. When they intertwine, they eventually are going to pull apart. When they pull apart, sometimes you get a flip-flop of genetic information. So these still have the same gene, right, which is represented by B, either capital B or lowercase b, depending upon the allele. What you'll notice is that those alleles have flip-flopped places. This process, when they get really close to each other, that's called crossing over. Note, they swapped chromosomes. This increases genetic variation, even more so, because originally your options were just a blue chromosome, just blue. But now you could pass on this blue chromosome, you could pass on this blue with a little bit of yellow, right? And the color is just representing that those genes have flip-flopped, right? They've flip-flopped tiny sections. The next way that you can wind up getting new differences, right? Inheritable variation or differences in the DNA is during replication. We have errors can occur. An error, remember, that's going to be called a mutation. A mutation is a change in the DNA sequence. That happens during replication. A replica is a copy, so replication just means copy. Although DNA replication is tightly regulated or controlled, and remarkably accurate, errors do occur and result in mutations, which are also a source of genetic variation. Oh my goodness. This one is so important because it's really part of the standards about that inheritable variation. Only mutations that are in the sex cell are heritable. So in order to pass a mutation down to your children, it has to be in your sperm or it has to be in your egg. Inheritable is one of those words that you probably don't use in your regular language every day, but this just means passed from one generation to the next. I listed a couple of types of mutations. Are you going to have to know these? I don't know. But the types of mutations could be a deletion, insertion, or substitution. Deletion is exactly what it sounds like. You've deleted one or more letters. Insertion means you've inserted one or more letters. And substitution could be a one-for-one, meaning you just change one letter to something else, or you could change a couple of letters. To give you an example, if I'm going to compare these, I've got C-A-T, C-C-C, that's different. If I keep following, I got A-A-C, A-A-C. I added that one in. When you add a base in, that's called insertion. I do the same thing for this one. C-A-C, T-A-A, different. I just changed one letter in this case, G to A. When you go G to A and you're just changing one letter, that's called a substitution. Next up, we have mutations caused by environmental factors. These are really just like what bad things can you personally do that will affect your genes. So we've got smoking, drinking, environmental toxins like pollution, things in the water that you drink, things in the air that you inhale, sunlight. Those things can all cause mutations. Those might not be heritable, but they also could be heritable. It just depends. Only mutations in the sex cells are heritable. So if you get skin cancer because you've been out in the sun a lot, or you've done tanning beds, that is not inheritable, right? That's because of an environmental factor. But if a mutation happens and it then affects your sperm or egg because of the environment, that's considered heritable. And our last section of the standard is genetic engineering. Genetic engineering is when humans alter the genetic makeup of an organism. Let's write that definition down. Humans alter. Alter means to change. The genetic makeup of an organism. They used to do a lot with this. This year they have or on the new test, they have mentioned CRISPR before. I'm just going to give you a few everyday examples. I do think whatever they give you will most likely be something that I've never discussed in class, and that you will have to use the evidence from the passage in order to answer. But I did see CRISPR mentioned once or twice. CRISPR is the actual technology that allows scientists to go in, find the gene of interest, cut it out, or edit it, right? So that's what CRISPR does. So this is when you can either insert or edit a gene that has a mutation. And they are currently doing that. We looked at the story about a girl who was going progressively blind due to a genetic disorder, and scientists working with dogs were actually able to edit the gene that was causing that malfunction in the vision. They then used that on the humans, and now those people are no longer going blind. They've done stuff like this with sickle cell anemia, where the cells are improperly shaped and they're due to a genetic mutation, and they're hoping to go in and then fix that issue, so therefore their red blood cells no longer have the wrong shape. An example they used to always use was human insulin. Are they going to use that again? I don't know. If you can't make insulin because you're diabetic, scientists can go in, they can take out the human gene that makes the insulin using enzymes. They're called restriction enzymes. It cuts out the gene of interest, and then they put that gene of interest into bacterial DNA. When they do that, that enables or allows the bacteria to make the human insulin. And then scientists can get the human insulin from the bacteria, and then diabetics have insulin. Over here, this is showing you an example of they want to make insect-resistant fruit. Bacteria have a gene that kill the insects. They remove that gene from the bacteria, and then they insert it into, this case, an apple, the apple's DNA. So they take some of the apple's DNA, they cut it, they insert the bacterial gene, and now this apple, when insects try to eat it, they will die. And then our last one is from that state lab. It says, apply concepts of statistics and probability to explain the variation in distribution of expressed traits within a population. This is going to be data linked. You're going to have to look at data, and you're going to have to determine if that data says it's a beneficial trait or not a beneficial trait. So variation, remember that means differences, and we're trying to see if that difference in your genes is good or bad. It's good if they survive. It's bad if they die. So here we have environmental pressure may determine if a trait is advantageous or not. Advantageous, again, maybe that's a word you know, maybe it's a word you don't know. Advantageous means, is it a positive? You'll need to analyze that to determine whether the allele is beneficial for that specific environment. The example we did was the lactose intolerance lab. And remember, the environmental pressure in this example, we had two environments that people were living in. People were either nomads or they were using agriculture. If you were a nomad, it didn't matter if you could drink cow's milk. If you lived on a farm, it was really good if you were able to drink cow's milk. Now, did you get that trait because you lived on a farm? Absolutely not. Why did you get it? It's because there was a mutation at some point. When you were a nomad, it didn't matter if you had that mutation. You survived, you didn't survive, whatever. But when you moved onto the farm and people were consuming milk, the ones who didn't have the good trait or the good mutation, they wound up feeling pretty crummy when they ingested milk products. The people who did have the good trait felt fine after they had milk products. So therefore, the people who were able to drink milk were more likely to survive because they had adequate food, and since they were more likely to survive, they were more likely to reproduce. So people who had the good gene were more likely to survive and reproduce. It does not mean that they magically got this trait, which is sometimes what people think. You don't magically get things. Mutations happen. Mutations are either good, bad, or neutral. If they're good, they will increase in number. Other examples that we looked at in class, because this is really going to link to our genetics, our evolution section. We looked at the rock pocket mice. They lived in the desert. Some of the desert was dark, some of the desert was light. Some of the mice were dark, some of the mice were light. Depending upon your environment, one of those variations was better than the other. Right? If you blended in, that was a good variation. You survived. Did you get that variation magically? No, you didn't get it because you needed it. You didn't get it because you wanted it. You got it because there was a mutation. Some of you had it, and then that wound up being better, so therefore you survived and reproduced. The other example we looked at was the beaks of finches. We watched that video that followed the Grants on that island, and they had the beaks that were different sizes, and during the drought, it was good to have a big beak, remember? Because you were able to crack open those seeds that were hard, and it was not good to have a tiny beak, because if you had a tiny beak and you were trying to crack open big seeds, you didn't have any luck there, and you died. And again, in the population, there was already some birds that had big beaks and some birds that had little beaks, and it just, sometimes some of them survived better than others. So this all had to do with food availability during the drought. And if you're not in my classes and you didn't see those, both of those videos, their videos, are based on Biointeractive. So if you go on that website, you'll be able to watch the videos so you can better understand what I'm talking about.